Am I too early?
You just don't, no, they're always late.
Oh, usually you're not here Monday this time, right? You get to stay.
Yeah, it's Monday, so.
Yeah.
You know, I don't like traffic, so no matter what, I'm here. I don't like traffic. Try to start that.
Oh, you're fine. You're fine.
But then she
I eat too much. At least twice. I'm not healthy. Okay, I didn't bring my notes, so I just thought I would check some. She does. Here we go. Any questions first? You are ready to go. It's, it's okay. Let's Well, I got done with the lab exam. I stayed up. I was here 4 hours yesterday. And I'm surprised nobody's here. I didn't see anybody. The library is open, but nobody's here.
The lab?
Sunday. Sunday. The library is open.
Oh, the library.
Yeah.
I thought she said the lab.
No, I was in the lab. I thought the lab did it. It took me 4 hours. It takes a long time to do the lab because the microscopes set up and all of those, make sure that everything's
Which
right.
slides did you set up?
I tell you in secret, precedence. I give it secret, okay?
Send me an email.
Yeah, I sent you what's the example. Be honest with you, as I told you, this title, how sneaky can I be? You already know what the questions are in the exam, right? If you go through the lab notes and you study them, you know what I ask about different types of blood cell, connective tissue. What else can I ask? I cannot play so much with this slide. That's how they are, Elastic fibers groups, elastic collagen fibers groups. Okay. Okay. Some of you, I don't think you know where the exam starts. The exam starts from cell cycle, right? And continues with the nervous tissue. So nervous function is not part of the exam, okay? So it's a 50 multiple choice and it's throughout all of the thing, all of the stuff that we have material. If I were you, I would look at the quizzes that I'm giving you in class that you can make sure you understand all of the answers, questions, So this question would be very similar to what quizzes we had. Yeah. No, we did cell junction. Cell junction was the first exam. Okay. There we go. Okay. I have question on mitosis. Make sure you know what is a prophase, metaphase, anaphase, and telophase. Okay? So, do you want me to go to, what do you want me to do? Do you have a question right now? We can start with your questions. Or you just want to give me a fast go through it? before
Fast
your
and
DA. You want to do fast to it? Okay. Okay, let me pass the attendance because I want to know who's in here. Okay, this is not for nothing is scared. I just want to see who's here. Okay, so when you study the interface, the phases of interface, they change in length. So make sure you know, like for example, G1 is variable, G2 is like, the different hours, 6 to 8 hours, mitosis only 1 to 2 hours. Things about the timing kind of important, so you have to know that, okay? And major events that happens in each cycle, like for example, G1 is protein synthesis, cell growth, okay? So know about that, okay? Like everything that is there. I mean, you told me to go through it. S for DNA synthesis, right? S DNA synthesis, and what this is important, why we have this synthesis happens. And I think there was a question on the quiz too. Okay, when I was a student, there was somebody who was a note taker for the class, and medical schools do. Anybody note taker here? Like takes notes for everyone and send it to everybody in the class? You don't have anybody like that? Why don't you choose one?
Just choose one.
No, I'm serious. It's a good idea. One person usually is the one that really is fast, take notes right there. That was medical school when I took classes. Anyway, I don't know, because it goes fast. I mean, you can still take your own notes, but there's somebody who does that, and it's like very efficiently do it. So 6 to 8 hours, okay. Then okay, GO cycle. What does GO mean? What phase is this? GO, like what? This is the vading phase. Cells go from G1 to GO and they stay there until they can go to the next step.
So would you not consider GO a checkpoint, or is it a checkpoint?
When the cell go to the from the G1, they go to the G2, there is a restriction point, the checkpoint. You can say kind of you can say that because they stay there for some time. Yeah, you can say it. You can also say, but for me, I just want you to know this is the cell, this is their phase that the cell exit there and they stay there. Some of them stay there forever. Some of them stay, but when they talk about restriction, police say G1 to G2. It's not like, yeah, just, they stay there and they go from G1. They go from G1 to GO. So you can also say it's a phase to check data, but I didn't go that specific, okay? No, the GO is like some neurons stay there forever in the GO state because they never, they don't divide. Muscles, cardiac muscles. Regulation, you have external factor, internal factor. Highlight that growth factor of regulation from external. They're outside controls, right? This was really like nutrients, how much nutrient is available. Now, for the internal regulation, I think cyclin is the one that is making a little problem with some of you. Cyclin, basically, these are the proteins that needed for the cell go from the G2 go to the mitosis, right? And they have to be bound, okay, to kinases. Kinases are enzymes that basically phosphorylate cyclin. So what I want you to know that I have draw a cycle for you. So this is important. You have to know this one, maturation promoting factor, which is called also mitotic cyclin dependent kinase, which is mitotic cyclin plus is kinase, is needed for the cell to enter the mitosis. Okay, this is the one that we want you to know. And this amount increases right, at the point when it's going to go from G2 to mitosis. This cyclin plus kinase increases in amount when the cell is ready to go into mitosis. If I were you, I highlight it. I haven't made the questions that is too detail oriented. Did it go away? Oh,
Well, your
I
microphone's
took this one. I took my microphone and I turned my voice on.
So MC, does that take place at the end of G2? Is that kind of where it
Yes,
takes place
yes,
before?
exactly. G2 to M, G to M. Sorry, I touched my face, but my voice went away, but my jacket was going away. Okay, so it's really, it's just, make it hard, because Dr. Sarko is talking about kinases, right? Phosphorylate, these are the enzymes that phosphorylate proteins. Like in this case, this one, phosphorylate cyclin and mitotic cyclin is just a kind of cyclin that the cell uses during mitosis. There are different types of cyclin. And these are all in your textbooks if you study your textbooks. Know the prophase, metaphase, anaphase, what is a telophase, what events happen, what is an anaphase. For example, sometimes when you read the notes are confusing. What is the main events that happen in metaphase? The main. Chromosomes migrate to equatorial plane. That's the main thing that happens, What is the main event that happens in the chromosome move to the opposite poles? Okay, so highlight those. And telophase is easy because it's going back to the interphase. What is cytokinesis? Where does cytokinesis completes? Telophase. It starts on anaphase, end of anaphase, but completion happens at telophase, okay? So cytokinesis telophase. Okay, what is apoptosis? Cell death. Okay, now extracellular matrix. Make sure that you know that what is a ground substance, what components makes up the extracellular matrix, right? You have to know fibers and different types of the fibers, and also remember this extracellular matrix has very important component which I told you. This is the juice, which was what? What was the juice? Ground substance. What is very important about the juice? The ground substance is bonds to what? What does it bond to? Water. Okay, that was a quiz question. Remember? Okay, good. Thank you. Somebody remembers. Thank you. Okay, so fibers, different types of the fiber. And do I ask questions from this list? Yes. So make sure you know it. Where do you find type 1? Where do you find type 2, type 3, type 4? All different forms have been asked. Not necessarily all, but they would. I have asked you about this, and that should be easy. This is the most dominant. Like 90% of all collagen fibers are type 1. So it asks you in the lecture and also the lab.
We spend a lot of time walking through the different elements of ground substance as far as knowing all the different reactions in the lags, gags, and
let me get there. Do I get there already? Okay, so let me just go through this, and I go there too. So reticular fibers, make sure they are gyrophilic, right? Are gyrophilic, they have affinity for silver stain. That's why you saw it when you saw reticular connective tissue. The fibers look, black, right? So make sure highlight that. Elastic fibers, remember, they are not made of adhesives. They are made of elastin. Okay, so that's the components. Highlight. See, elastic, elastin. These are all components of the extracellular matrix. Now, for ground substance, you have to know what is glucose, aminoglycans are. What? The polysaccharide gags or polysaccharides. The linear polysaccharide, form. No, I haven't asked you what is. That's what you're asking. No, I haven't asked you the name of this. No. I hope you have studied by now, but no, I haven't asked you. Just know these are linear polysaccharide, and they are attached to the proteins, and you make a proteoglycans. okay? So the components of the ground substance is lots of gas. Also, you have gas which are attached to the protein which make up proteoglycan, but you could have just gas, okay? And they're attached to, this makes them hydrophilic, so highlight that. This is important to understand that they are hydrophilics because of presence of sulfate and carboxyl groups. Okay? Do you remember I showed you the pictures, those awesome pictures, beautiful pictures, green, red, all that. Remember adhesive glycoproteins are what? Like fibronectin you study, and you see laminin, you see the integrins. Make sure what they bind to, okay? And I don't want to just read them for you, okay? Make sure what they bind to. Do you understand what I'm saying? For example, if I ask you, it says like fibronectin, okay? First of all, remember fibronectin most probably is formed by what cells? Fibroblasts, okay? Fibroblasts are cells of connective tissue. What do they secrete?
Collagen.
Collagen. I hope you know that. Fibroblast. Fiber, collagen. Blast. Blossomy. Okay? So fibronectin, for example, you should know, okay, bonds to cell, collagen, and heparin. The thing is, some of you maybe have studied this 100 times. You just memorize point by point. Seriously. But when it comes to the form of questions, oh my God, where was that? Because you didn't understand it. Okay, so make sure that you check it with somebody, you study with somebody, and question each other, because you just memorize it, and then when it comes to the multiple choice, you don't know which one is right. Okay, also laminin. Laminin is like if I ask you secreted by what or synthesized by what, you should know by epithelial cells. Okay, so they're just, what does it bind? Collagen, heparin cells. And also remember, laminin, if you read here, also bonds to integrins. The collagen that it bonds is type 4. Now, some of you memorize collagen, but if you don't remember, if I ask you, if I put collagen type 4, you don't remember, right? Which collagen was, so, okay? Okay, now we're doing so good now. Okay, mesenchyme. No back and forth about mesenchyme. Okay, so this is embryonic connective tissue. Where do you find it in that three germ layers? Mesoderm. They are pluripotent cells. What does pluripotent mean?
They can become different things.
Yeah, they have the potential. This is potent. Polar means several. They can become many cells of the connective tissue. They can become bone cells, fat cells, all different cells. Make sure that you look at also the slide of mesenchyme, mucoid or mucose or Wharton's jelly, because whatever I'm saying right now is true for lab exams. So make sure you look at it and you can distinguish between them. The different types of adult connective tissue, what they are, we have loose and dense, okay? Special, let's see. Functions, I assume that should be very straightforward. Phagocytosis. You know what phagocytosis is, right?
Engulfing.
Engulfing the bacteria, yeah. So components of connective tissue. Okay, make sure you know it. Sublimate, right? Cells, fibers, juice. Now, if I ask you, elastic connective tissue, it has this component, right? So all connective tissue share this, okay? Now, which one are fixed cells? Which one is which cells are fixed, which cells are free. You have to know, that's really obvious. Fibroblast, make sure you know which one is active, which one is inactive ones, I think you have the slides that we have. Adipocytes, make sure you know, okay, I haven't got that, but there are two different types, adipocyte, right? Unilocular and multilocular. One has one single droplet of fat, the other has several, right? Okay, where do you find reticular cells? Lymphoid tissue, you find it in the bone marrow, lymphoid tissue, Also have asked questions like that, where do you find them? Okay? Macrophages, no back and forth but macro. I have some affair with macrophages. So no back and forth but what it says, right? I have given you like matching, so you have to know, all of these things here. What cell secret is a matching type of question. So it's not all multiple choice. Does that make sense? Just like the one I gave you when I gave you the quiz. Okay? So for example, if I ask you Langerhans cells, don't just say, oh my God, what is Langerhans cells? These are what? What are Langerhans cells? Macrophages. These are macrophages, just the name is different. All of the blood cells are coming from where in the body? The adult. Bone marrow. These are all derived from bone marrow. All blood cells are derived from bone marrow. All blood cells are derived from bone marrow, remember that. So if I ask you macroglial cells, these are macrophages, and what is the function? Phagocytosis. Yeah,
So you're not
they found. Oh yeah, I will get into nervous tissue. I asked there too. So make sure that you know. Mast cells. There is what? You just have to memorize heparin, histamine. These are for allergy. These are the ones that have IgE receptors. So they are important, very important. So make sure that you know that. Where do you find the mast cells? In the connective tissue of the dermis, digestive, and respiratory. Where basically the body needs them to be there. What is it specific about plasma cells? Where should I highlight? If somebody tell me highlight somewhere, I would highlight this. So, go find the stuff that is, all of this important, derived from the lymphocyte, central nucleus with distinct peptide. These two mostly would be histology, they derive from the lymphocyte. Okay, they give rise, B lymphocyte give rise to what immunity? What type of immunity coming from B lymphocyte? Which one? It says here. Antibody-mediated immunity. Okay? Because these are B cells. T cells give cell-mediated immunity. Right?
They're what?
Okay, I get to the bones. I get to the blood cells. So for now, just write B cells produce plasma cells. Plasma cells produce or secrete antibodies. Okay, I get to that. Make sure you know what you find in the mucoid connective tissue, which is mucoid connective tissue is found where in this on the umbilical cord. Remember this slide that you see? Make sure you go revisit it again. Remember mucoid slide that you see? Mucoid connective tissue. Make sure you look at the fibroblasts and collagen fibers. Reticular, this is the easy one. So if you look at reticular connective tissue, what type of the fibers you're supposed to see a lot of them? Reticular fibers. Or I can ask you type 3 collagen fibers. Okay? So these are interchangeable. Reticular fibers, these are actually collagen fibers. Okay, they're type 3. Now, these reticular cells are kind of fibroblasts. They're modified fibroblasts. Because remember, fibroblasts are the ones that secrete collagen. Now, reticular cells are actually modified fibroblasts. Okay? Okay, notice the fat cells. What is multilocular cells? Does it produce white fat or brown fat?
Brown,
Brown fat. Well, how about unilocular?
white.
Good job, you learn about this. Okay. This irregular connective tissue, where do you find it?
And
The skin, good job, the skin, organ capsule. This is the most dominant type of connective tissue after loose connective tissue, okay? That's the most, because you see it around the capsules, periosteum, perichondrium, and make sure how the fibers are arranged. They're very stress resistant. This presence of collagen in the organs are very important, okay? Because it gives a tensile strength. So if you put collagen and next to collagen, write this down, it gives a tensile strength to the organs. It's very strong. These are like the tension resistant, okay? Collagen fibers. I'm giving you the best review I've ever given. Okay, this regular connective tissue, tendons, and ligaments, you should know. I try to ask questions that are not only important as chiropractor, but also the one that I think board would ask you for the exams. Okay? Elastic, where do you find it? These are again type 1 collagen fibers. So don't get confused. If I have elastic connective tissue, I have elastic fiber, but I also have collagen fibers. Okay? So, but majority I would assume it would be elastic because that's how you name it.
Question.
Yes.
I remember you also had elastic fibers having good tensile strength.
No, collagen is the most. If what do you mean? Yeah, elastic fiber have the ability of stretching. You can stretch it and they go back to fiber. But if they ever say what is the strongest fiber, the most, the greatest tensile, the word is tensile. You will say collagen fibers from now on. Collagens are the strongest and the most dominant proteins in the body. Okay, remember that. Okay, now cartilage. I asked a lot of chondrocyte, chondroblast, lacunae, all this good stuff I have asked because they are important. Okay, make sure to know the specific characteristics of cartilage and also bones. Since I'm at the bones, I'm going to go talk about the bones too. But they lack nerve endings. Lacks vasculature, lacks lymphatics. Okay. Now the bones are opposite. They have nerve endings and they are very vascularized. Okay. So make sure you know the differences between major differences between bone and cartilage. Make sure you know where you find hyaline cartilage in the body. Is the most dominant type of cartilage in the body. Hyaline, what type of fibers you find in the hyaline cartilage? What is the most dominant? What is type? Which one? Type 2. Type 2. Good job. Perichondrium is important type of the connective tissue that is made of. This is regular, and this is the one that provides nutrients to the cell. And this is the way the inner layer of the producer is called chondrogenic zone. That causes the interstitial growth of the cartilage. Okay, cells, no chondroblast, no chondrocyte. You see here, chondroblasts reside in lacunae. Chondrocyte also reside in lacunae. Okay? It's different than osteoblast and osteocyte because osteoblasts, they secrete this material in themselves, and once they stuck in that lacunae, they're always in the lacunae. Osteoblasts do not live in lacunae. Let's put it that way. Yes?
So you sit in the chondrogenic zone. I thought it was appositional.
Oppositional growth, yeah, I would just think as an
I
opposition,
was like,
yeah.
you said
It
interstitial.
goes,
when
you know,
I
as the diameter increases.
was- Okay.
Interstitial growth from inside out. Thank you for catching me. So do you highlight this? Chondroblast reside in the cone, reside in the cone. Now, if I ask you, osteocyte always reside in the cone, not osteoblast. Okay? Osteoblasts live on the surfaces of the bones. Remember? Okay, okay, no, the ground substance, I need to finish up. Nutrition, okay. Now, remember hyaline cartilage, the articulating cartilage does not have precondylium, so highlight that. You get confused. This type, that's why you have synovial fluid on the joints. So highlight that, that's the nutrition, okay? Now, elastic cartilage, does it have perichondrium? Yes, okay? How about, and make sure you know where you find it, make sure it's the type 2. How about phagocartilage? No, okay, so highlight that because the student get confused, okay? It does not have, you know what perichondrium means? I'm going to talk to myself. You know what is it, right? It's a dense, irregular connective tissue. Because don't get confused when you see on the exam, oh my God, I thought all of them have perichondrium. Not all of them. Okay, perichondrium is missing the shadow cartilage and also articulating cartilage. Make sure you read where they are found, okay? Okay, so as she corrected me, interstitial growth is different than appositional growth. Okay, interstitial growth is growth from inside out. Okay, so when you have chondroblast divide and they become many and isogenous groups that are the same. Appositional growth, when you grow from the inside the precondylium, they grow. Hopefully you have a study. These are important, interstitial and appositional growth. Okay, bones, no back and forth. I made a lot of questions on bones. As you can realize, it's kind of important for us. So I asked a lot of questions on the bones. Osteoblast, osteocyte, everything, vascularized. It has also nerve endings. What is the function? Calcium deposit, okay? Sorry, I'm going fast. I need to finish this at 45. The type of the bone, and make sure you look at histology to decalcified bone, ground bone, and also models of as from the bones. Sharpie fibers. Stop me if you want to. Okay. Osteoblasts, make sure you know. These are the, if I ask you which one would secrete or provide organic matrix, and also calcification also done with osteoblasts. So, and they remember that is formed by osteoblast. Make sure you know what is an osteoid. What is an osteoid? Osteoid is not calcified yet, right? So osteoid, it has type 1 collagen ground substance, so it doesn't have the calcium phosphate deposits yet. Okay, so that's a big distinction between osteoid and the bone matrix when we talk about that. And osteoblast give rise to osteocyte. Okay. Okay, osteocyte maintained, highlight this, maintain bone matrix. Osteoclast are resolved bone. So if the person has osteoporosis, which one is activated? Osteoclast. Osteoclast is the one that causes the pores, right? Osteoporosis means bone that has many pores, like holes. Okay? So make sure, if you don't know anything about this exam, make sure you study these 3 types of the bone cell. And these are all coming from osteoprogenitor cell. again, you have to know the hormonals, parathyroid hormone, calcitonin. What does parathyroid does? What does calcitonin do? Parathyroid is coming from what gland? Like this one does not say from what gland, but you have the cover points. Parathyroid hormone is coming from parathyroid gland, right? This one, calcitonin, is coming from thyroid. So calcitonin activate osteoblasts, makes bone, right? Parathyroid activate osteoclasts. Make sure the combination of the matrix, you have organic, inorganic, you have collagen fibers that gives the flexibility to the bone, and we have organic that gives the hardness to the bone. The hardness of the bone is coming from this deposit, peroxyapatite crystals, calcium and phosphate, calcium carbonate, calcium, So calcium phosphate deposit is good for you. Okay? Now, make sure about all this osteon, what is a concentric lamellae, you have Volksmann canal, spongy bones. What is the intramembranous bone? It's coming from what? What cells that differentiate and they start the formation of intra? Mesenchyme. So you have mesenchymal cell. Mesenchymal cell differentiate, they become osteoprogenitor and then osteoblast. So highlight how did you get there, okay? So know the process of formation. Endochondral calcification, you saw it one in the, you saw it's the most bones of the body, but this part here, rectangular bone formation. Remember this zone of reserve, zone of proliferation, zone of hypertrophy? This is all endochondral calcification, right? Because the cartilage is becoming bone. So make sure you know it both for the lecture and also the lab. Woven bone, what is woven bone and when it forms? Okay? Is the first, good job, highlight that. So always you have woven bone forms which is, which is low in mineral, high in stocyte, irregular orientation, all that. How about in the injury? Does woven bone forms? Yes. Yes. Always you have woven bone before intramembranous or endochondral. So write this down. First bone formed by either of the above method. Nutrition, blood. Okay, I have asked back and forth about plasma, what are the components of plasma. I talked about red blood cells. You have to know red blood cells are, what is the life cycle of red blood cells? How long did they live? 120 days. It's not in your lecture notes, but I have told you, right? Because it was in my board exam, so know it. 120 days, which would be how many months? 4 months. Good job. What is the most dominant protein in the blood? Albumin, right? So you have this in your PowerPoint. So study that. You have this. It's not there, but in your PowerPoints, in your books, and all that. Red blood cells. Know the structure. Know about the hemoglobin. Know about oxyhemoglobin is when the hemoglobin bonds to oxygen. These notes you see, they're very summarized. If you just memorize, I don't know how you can understand this if you just memorize it. You said the oxyhemoglobin bond to oxygen. What? This is hemoglobin. It's talking about hemoglobin that's bound to oxygen and makes oxyhemoglobin. And is this reversible? Yes, it's reversible. How about carbaminohemine bond to carbon? This is hemoglobin that bonds to carbon dioxide. Is this reversible? Yes, and it's called carbamine. How about this one? Carboxy Hemoglobin, this is carbon monoxide poisoning, basically, it's irreversible. You know, I have asked many questions, I don't even go through it, because I have asked lots of questions about eosinophil basic pills. You have pictures in the lab, you have, make sure you know which one is granulocyte, which one agranulocyte. So make the distinctions between them. You should know which one is the highest in granulocyte number, which one is the lowest, all that. Do you know what I'm talking about? Okay. Minutes. Monocyte, the largest one, and you know. Platelets, what is the one that big cells that become platelets, you should know is the megakaryocytes. Megakaryocytes, okay?
What were you saying for B cells is
Oh, I haven't got it. Where is
that
this?
type of immunity,
Lipos.
and then the T
Good.
cells.
Thank you so much for reminding me. Oh my God, thank you. Yeah, make sure you know that, okay? When I talk about lymphocyte, remember lymphocyte? First of all, these are the highest number of agranulocyte, the highest number, right? Okay? And then they're divided into two groups. We have B cells and T cells. You should know B cells, okay? B cells are the one that differentiate into plasma cells, we talk about, right? And plasma cells give rise to antibodies. And this type of immunity is called humoral immunity, or the other one that I said, what was the name of it? Antibody mediated, because in this type, antibodies travels throughout the blood and they go throughout the pathogens. So it's different than the other types. All of these B cells, they are all lymphocytes, these are all starts from the bone marrow. They don't go to the thymus. Now, T cells are also starts from bone marrow, but they go to the thymus organ or thymus gland and they become mature there. So this is the place that the maturation, they become complete, they become T cells. Now, the way T cells do the specific immunity is by the system of immunity that we call it CMI, cell-mediated immunity. They form T helper cells, cytotoxic T cells, and they kill the cells, okay? It's not like here, when you produce the antibody travels in the blood. Humoral means what? We talk about humoral, means fluid. Okay, like blood is fluid, right? Lymph is fluid. They travel in the, so humoral immunity is fluid immunity, like they travel in blood. This system, T cell mediated immunity, they produce T cell and they produce the different type of cytotoxic helpers, and they all go and cytotoxic cell goes and basically kill infected cells, okay, tumor cells. They kill cells. Cells kill cells, okay? I'll spend too much here. Okay, nervous tissue. Oh my God, this is the love of my life. So I have asked a lot of questions. This is cool stuff. This nervous tissue. You should know what when you talk about different types of the neurons. Make sure you know first of all the characteristics of the neuron. We have a sensory, we have motor, we have interneurons. Interneurons, or what is another name for interneurons? I just touched on Thursday. What? Association. Good job. Association. Okay. So go through the component. What is the Nissl substance or Nissl body and Nissl substance are the same. These are polyribosome or rough endoplasmic reticulum. They call it. You find them in the cell body. I'm sorry. I know I'm going too fast, but I don't have a choice. So study that, make sure you can distinguish between different parts. What is the axoplasm, axolemma, axonida. Dendrites, make sure you know what is a bipolar. Simple definition will do it. What is a pseudounipolar? Where is the collection of pseudopolar neuron cell body you find? Where is the collection? Where? Dorsal root ganglion. Okay, so write this down. Pseudo-unipolar cell bodies, the collections of them, are found in DRG, dorsal root ganglion. That's why dorsal root ganglion is sensory, because these are sensory. Okay? So pseudo-unipolar, sometimes they call it as a T. They look like a T, meaning they have a little process that divides. Remember, they have a prefrontal process and central process. What is a multipolar? What is the most dominant type of the neurons? Are they unipolar or pseudo-unipolar or multipolar? It's right there. Oh gosh, I'm so smart. I didn't see that. I was gonna ask you something now. Okay. And multipolar are usually what? Are they sensory or motor?
Motor.
Okay, synapses, electrical, chemical synapses. I will talk about it on Tuesday, on Wednesday when we start talking about nervous function. Okay, now supporting cells of the central nervous. I have asked a lot of questions, so you should know that. Okay.
Say again?
I have asked a lot of questions on them, like, okay, what are macroglial cells? These are astrocyte, oligodendrocyte. What are astrocyte are good for? Okay, sure, read this, all of it, because these are important, and I see their importance for us as chiropractors. I keep asking questions. Oligodendrocyte, what do they do? Macroglia, phagocytic. Are they macrophages? Yes, right? Macroglia are macrophages because they are for phagocytosis. Okay? Ependymal cells, what do they do? Schwann cells, what do they do? What are the differences between Schwann cell and oligodendrocytes? Okay? People just arriving. Famous bagel. No, not famous bagel. It's not called. Famous foods. Ishmael, lantern, and plaque. Make sure you look at the histology of this thing. It's easy, but some of you don't know of Randy. What is the myelin is good for? Satellite cells. Do I find satellite cells in PNS or CNS?
PNS.
Yeah. Autonomic division. What is the central nervous system? Brain, peripheral nervous system. And, don't get confused because students somehow they have issues with choosing cranial nerves in the peripheral nervous system. But believe it, cranial nerves are part of the PNS. They are not part of the CNS. I mean, you can choose it on the exam as cranial nervous system, but you got the wrong number. You get wrong points. I mean, seriously, it's right there. So, no, don't choose the wrong option. I'm telling you that.
What's the wrong
I want you to know it because you are nerve doctors, so this is like the this is the entry to learning more about this nervous system. So make sure you know which one is. These are the basics, and then functional division, somatic, inner breast, skeletal muscle, autonomic nervous system, smooth muscle, cardiac gland, and then white. I have asked questions a lot. What is the white matter? You should know collections. Myelinated, unmyelinated, gray matter. Make sure you know cerebral cortex. What did you see in the cerebral cortex in the lab? They don't have it here for pyramidal cells, right? In the cerebral cortex, make sure check them out. Cerebellar cortex, again, these three layers, you should know back and forth histology of them, and also, fine motor and posture. Start reading about this. So this is just the beginning. You have to know a lot of cerebellum later on. The spinal cord, I have given you a question. I think ventral horn, what is the dorsal horn, central canal, no histology. Hey, good morning. Peripheral nerves, we did add. Okay. Epineurium, perineurium, endoneurium. Make sure you know the differences. What is what? Because I feel like you still don't know it. And also for the muscle, remember, muscle histology, okay? Ganglion, collections of neuron cell body outside the CNS, okay? That's what we call a dorsal root ganglion. Make sure you go and check your histology of dorsal root ganglion, how it looks like, parasympathetic ganglion, how it looks like. Parasympathetic is the easiest one. Why? Why is the easiest one? Because it's located within the muscle layer, right? In the digestive wall. In the walls of organs, digestive organs. It doesn't have a capsule. I'm sorry, I'm going too fast, but this is it. And then sympathetic. Make sure that you look at the histology of this. These are multiple motor neurons. These are ganglion, right? Because ganglion is connection of the cell bodies. Okay, good job, guys. I'm glad we made it. We all made it through. So give me the attendance.